Hi everybody. We're getting close to the end of our macromolecule study here, talking about proteins in this video, and I have a confession to make. I think proteins are absolutely fascinating, and I could talk to you about proteins for the whole rest of the summer and still not run out of things to say, because I just think proteins are that important and that interesting. My guess is you don't think proteins are that interesting, so I'm going to try really hard to limit what I tell you to what you absolutely need to know. It's going to be a bit painful for me, but here we go. By the end of this video, we're going to be able to describe the function and the structure of proteins, and then we're going to understand how a special group of proteins called enzymes work. So the polymer of amino acids is called proteins. The monomer of a protein is called an amino acid. Amino acids are the individual beads that make up the chain. The protein is the whole necklace. So let's kind of deconstruct an amino acid here and look at all of the different parts and pieces. Okay, so starting here in the center, we have something called the central carbon. Sometimes you'll see it as the alpha carbon. It's in the middle, and it always has four things attached to it. Okay, so on one side, you're going to find an amine group. It's going to be a nitrogen with two hydrogens. Opposite that, on the other side of the central carbon, will be a carboxyl group. So it's a hydroxyl plus a double bonded oxygen. Between those two, you will always have a hydrogen and also an R group. Now, I'm going to tell you an embarrassing story. My freshman chemistry class, I remember spending a very long time, an embarrassingly long time, in fact, searching on the periodic table for an element that started with R. There isn't one. R is kind of a stand-in code for any group of molecules that gets attached at the fourth side of carbon. Okay, so when you look at an amino acid, you should be able to find the amino group, the carboxyl group, the hydrogen, and something else. And that something else is the R group. And there are more than 20 different R groups. So here is one R group attached to the carbon here, central carbon, carboxyl group, amino group, hydrogen, R group. And this particular chain in that fourth place makes an amino acid called methionine. When we have a different chain over here in the fourth spot on the carbon, we have an amino acid called cysteine. This chain with the ring structure is tyrosine. This chain, a bit more complex, is arginine. Okay, so we can plug in a wide variety of carbon-based chains into the fourth place on the amino acid, and that's going to give the amino acid lots of different properties and ultimately influence how the amino acid is able to fold into the overall protein. So a peptide is any time two amino acids or more than two amino acids are joined together, and the specific bond that forms between amino acids is called a peptide bond. Okay, if we just have one amino acid, that's the monomer, it's just an amino acid. If we have two amino acids joined together, that's a dipeptide. Three amino acids joined together, tripeptide. 10 to 15 and more than 15 oligo and polypeptides. The numbering here is a little different depending on what source you look at. Now, a full protein can be more than 1,000 or multiple thousand amino acids long. So this is how we would describe very short chains of amino acids. We form peptide bonds by dehydration synthesis, just like we saw in forming a triglyceride, just like we saw in forming disaccharides. It's the same exact thing. We have the OH from one and the H from the other. We pull them off and make them into a water, and we join the remaining molecules together. So here they are joined together, the carbon double bond to oxygen, carbon double bond to oxygen, gets joined together to the NH from the amino group over here. Dehydration synthesis, we're putting it together by removing a water. Conformation of a protein is absolutely critical to that protein working. The protein's shape is super specific. The three-dimensional shape that a protein takes is very complicated, and if it takes the wrong shape, it doesn't work. There are things that can denature a protein, and when I say denature, I mean change the structure of the protein so radically that it cannot recover. Small changes in a protein, generally, they can kind of go back into the right shape. Big changes, they're just ruined. So heat and pH are the two ways that we can denature things. Either intentionally or unintentionally. If you don't like raw eggs, you want to denature the proteins in your eggs before you eat it by cooking it. I know there's a lot of text on this slide. I wanted to talk about all four structures together. I think it's important that you kind of see the whole picture here. So we talked about amino acids getting put together one after another in a straight line to make the polypeptide. That polypeptide, the specific sequence of the amino acids, is the primary structure, and it's coded for in the genes of whatever organism it is. Okay, so the keratin that makes my hair is coded for in my DNA. Now, my sister's hair is a little bit different. It's a different color. It's a different texture. It's not wavy. That is coded for in her DNA, and those characteristics come from the primary structure of the keratin that makes up her hair. Secondary structure is when we see these two very common repeating things, alpha helix and beta helix. And we're going to see a picture of these in a second. And these come from a sequence of amino acids that has a specific pattern. So these are kind of underlying structures that make up what we call the secondary structure. I think that'll make more sense with the picture on the next slide. The tertiary structure is the three-dimensional shape that the protein takes as it's getting ready to become functional. It takes the three-dimensional shape based on stability. It wants all of the hydrophobic amino acids with their hydrophobic R groups to be all grouped together on the inside of the protein, and it wants all the hydrophilic amino acids that are happy to interact with water on the outside of the protein, and this is all stabilized by a lot of chemical bonding that goes on within the structure. So that's the tertiary structure. And then finally, we have something called the quaternary structure, and the quaternary structure is found in most proteins, but not all. So sometimes you have 2 or 3 or 4 or even more polypeptides with their full tertiary structure that have to come together in order to make a functional protein. So if we talk about hemoglobin, hemoglobin is the protein that carries oxygen in your blood. Hemoglobin has a quaternary structure. It's made of 4 peptides. Those 4 peptides come together to be able to carry your oxygen and circulate with your red blood cells. Okay, so hemoglobin has 4 polypeptides. Each one of those polypeptides has its own tertiary structure, its primary structure, and then its sequence of amino acids that is coded for in the genes. So looking at a visual of this, we have the primary structure, the sequence of amino acids, what order they're lined up in. We're going to talk a lot more about this in a later section. Then we have the secondary structure. So we have the alpha helix, and sometimes this is called a beta helix. Sometimes it's called a beta pleated sheet. I prefer the pleated sheet Pleated means like accordion fold. So we have like an accordion folded piece of paper here. And you'll see these repeating patterns. It looks like we have an alpha helix here, alpha helix here, alpha helix here, and then sections where we have the beta pleated sheet. And this is the tertiary structure. This structure is stabilized by bonding inside between different portions of the polypeptide, and also wanting to keep the hydrophobic parts away from water and have the hydrophilic parts interacting with water on the outside. And then we have several different polypeptides coming together to form the functional protein that can go out and do whatever job it's going to be doing. Proteins have a ton of different functions. I'm not going to read these slides to you. You are very capable of reading them on your own. They are structural, communicative. They do transport across membranes into and out of cells. They are catalysts, and we're going to be finishing up this video by talking about how those catalysts work. They also do recognition. They also work in your immune system to protect you from foreign invaders. There are some that are able to move the entire cell, and there are some that bind things together and keep them from coming apart. Enzymes. Okay, enzymes are one group of proteins that serve as a catalyst, and a catalyst, if you recall, is anything that makes a reaction go faster. The substrate is the thing that's coming to the enzyme that the reaction is going to happen to. So the substrate is what we start with before. Okay, we have another naming thing here. Most enzymes end with ASE. Okay, remember that most sugars end with OSE. Most enzymes end with ASE. This one's pretty important. This can give you some big clues if you're trying to figure out what something is. The way that an enzyme works is they lower the activation energy needed to get the reaction started. Okay, this right here, this is a key. I would fully expect to see this on an exam. I think this is critically important information. I think you need to know this. I think you shouldn't pass a biology class if you don't know this. So I would really make sure that you take a good look at this before your exam. Enzymes function by lowering the activation energy that we need to get the whole process started. I'm going to go out of order here. Let's talk about this one next. Okay, so let's talk about how the enzyme works, and then we'll go back and we'll look at how it influences energy. So the enzyme is shown here in blue, and it has something called an active site. Okay, so here's my active site. And here's my substrate. The substrate is the unreacted thing. It's the starting product. And my starting product is going, or my starting reactant is going to come into the active site. Can you guess what this thing is? Do you remember from the carbohydrate video? This looks like a 6-sided sugar joined to a 5-sided sugar. This looks like a pretty accurate representation of sucrose, a glucose joined to a fructose. Notice that the active site is really similar in shape to this. If we had mannose, a glucose and a glucose, try to fit in here, it ain't gonna fit. All right, so the active site is very specific to the substrate. So the substrate is gonna come into the active site, and the enzyme is actually gonna change shape around it and hold that enzyme in place, or hold that substrate in place while the reaction happens. And this reaction is a catabolic reaction. We're going to break those two pieces apart, and we're left with product 1. We're guessing that's a glucose, and product 2 are fructose, and they're going to go on their separate way. And this is also a key. This enzyme right here is exactly the same as the enzyme we started with. Okay, the enzyme is a tool. The enzyme works to separate the substrate, in this case. Enzymes can also join things together, but in this case we're separating. But the enzyme, nothing happens to it. It's ready right now to take on a new sucrose and repeat the process all over again. Okay, so let's go through this one more time. We have the enzyme. The active site is free. The substrate comes into the active site. It's a very specific fit. The enzyme is going to hug into the substrate, so it holds it in place, and that helps the reaction, separating these two things, be more efficient. Once they're separated, product 1 and product 2 are free to do their own things, and the enzyme is ready for another substrate to come in and start the process all over again. All right, now let's talk about the activation energy part of this. Okay, in order to go from our substrate to our products, we are releasing energy. Okay, so we've got more energy at the beginning, less energy at the end. The energy had to go somewhere, it goes out. In this case, it's going to be released probably as heat. Okay, keeping your body warm. It takes a lot of energy to keep your body at 98.6 degrees. So we're starting with some energy and we're ending with less energy. But we have this thing in the middle. It's called the activation energy. This is the energy that it takes to get everything set up for the reaction to start. This is a pretty high amount of energy, and the cell needs to get it from somewhere. And sometimes cells can, and other times without the enzyme, the cell cannot do the reaction. Here, where we have the reaction present, where we have our catalyst present, we still have the same amount of energy at the beginning, we still have the same amount of energy at the end, but we have lowered the activation energy. We have lowered the amount of energy that the cell has to invest in order to get the reaction started. So this is much more efficient for the cell to do. Couple key points here. Enzymes are reusable. They're the same at the beginning as they are at the end, so they can be used over and over and over, sometimes thousands of times before they start to degrade and have to be replaced. There are some enzymes that can catalyze a reaction 1,000,000 times per minute. It's absolutely incredible. Enzymes are sensitive to both temperature and pH, and most enzymes work in the place that has the proper environment for them. Okay, so there's an enzyme called amylase. It's found in your saliva. It works best at pH 7. Guess what pH the saliva in your mouth is? It's generally pH of 7. Pepsin is an enzyme that works in your stomach. Pepsin's perfect pH is 2.0. If we tried to put amylase into your stomach, it wouldn't work anymore. If we tried to put pepsin into your mouth, it would not work. Also, pointing out here, do you guys see ACE at the end here, giving you a big clue that it's an enzyme if I wasn't already telling you that? Most of the enzymes that humans have their perfect operating temperature at 98.6 degrees Fahrenheit, which equals 37 degrees Celsius. All right, guys, this is why fevers are so dangerous, especially for little babies. What a fever does is, it's trying to kill any foreign invader pathogen that has taken over your body. But in the process, it can damage enzymes. If you have a very high fever and your enzymes stop working or even work less efficiently, that could slow down your metabolism and all sorts of reactions. So little babies who are still, their biochemistry is still working on making some enzymes for the very first time, when they have a high fever above 105 degrees, it can really damage their enzymes and cause a lot of body functions to stop. So that's a little bit why fevers are so dangerous. Cofactors and coenzymes. Cofactors are generally some sort of metal. Coenzymes are a vitamin. Okay, cofactors are a metal, coenzymes are a vitamin. These are extra bits that the enzyme needs to work. A really famous one is iron is needed by hemoglobin in order to carry oxygen. So the iron would be a cofactor because it's a metal. And that's cofactors and coenzymes.